Well, hey, everybody, this is Robert, and welcome to today's show. Now, Naegleria fowleri is a microscopic amoeba, which is a single celled living organism. It can cause a rare and devastating infection of the brain called primary amebic meningoencephalitis, or also known as PAM. The amoeba is commonly found in warm freshwater like lakes and rivers and ponds. Now, infections can happen when contaminated water enters the body through the nose. And once the amoeba enters the nose, it travels to the brain where it causes PAM and the destruction of the brain tissue ultimately. And it's typically way more than not fatal. So, how does the body's immune system react to this parasite? And what research is being done on the immune response? Well, joining me today is Ashley Moseman, PhD. Dr. Moseman is an immunologist at Duke University, and his lab is studying the host immune response to Naegleria fowleri. Dr. Moseman, welcome to the show, sir.
Thanks a lot, Robert.
So before we get into some of the immunology type questions, I did say in the intro how people get, how they contract the parasite, and this really is a precursor to the immunology questions. Can you detail how an individual actually contracts this?
Well, I think it's presumed how this happens for the most part. We model all these things in animals. I don't work with humans, but what's presumed is that the amoeba somehow gets into the upper airway of your nose, essentially. So you take a lot of water up the nose, and it's contaminated with amoeba. It gets into these upper recesses of your sinuses, where your olfactory surface is, the part that you use to smell. And from there, it can kind of latch on and cross over into the olfactory tissue, and it follows that olfactory tissue up into your brain. And that's where the problem really begins. But yeah, that's essentially what is believed to how it happens. It goes up into your nose, crosses your smell surface, and goes right up into your brain.
Yeah. Now, I've talked about this amoeba many times on the show, but today I want to look at it from a different angle, the body's immune response. And before we get into that, I want to talk about, or at least get some definitions for the audience about immunity. Dr. Moseman, what is innate immunity and what is adaptive immunity?
Yeah, so innate immunity these are obviously very broad topics for us as immunologists, but innate immunity basically just is talking about things that will immediately be in effect. So if you get stung by a bee and you start getting swelling right there, right away, that's an innate response, something that happens immediately. You don't need time to develop it. It just kind of happens. It can kind of get bigger and bigger and bigger over time. These innate responses continue, but it doesn't take a lot of time to educate your immune system to get it to do something. It just does it. It has these patterns that it recognizes from along millions and millions of years of evolution that just go into effect immediately. And then adaptive immunity is more nuanced. It takes longer to elicit, so it's much more specific. So whereas certain innate immune cells can tell, oh my gosh, there's a bacteria in here, your adaptive immune system will be able to say, I know exactly what kind of bacteria and exactly what shape on the surface of that bacteria I want to respond to. And so it's very, very specific in the innate and the adaptive compartment, but it takes much longer comparatively to get that adaptive immune response. So you're thinking about for innate, it's like minutes to over the first few days of a response, and adaptive really starts to come in after like 5 days or more. And the adaptive immune response is really what you take advantage of when you do vaccines. So you want to get something that's very specific to a virus or whatever you're immunized against, but it takes some time to educate your immune system to get the adaptive immune response to do that. But once you have it educated, it's extremely powerful and can basically prevent you from getting sick in many cases.
Now, I understand there's many open questions regarding this topic today, and I'm going to give you some time later to talk about the research that you're doing. But how do each of these types of immunity play a role when dealing with Naegleria fowleri, as far as you can tell?
Yeah.
And does the immune response contribute to its pathogenicity?
Okay, so those are many questions, but yeah, I think in general, again, in humans, I think it's probably not well understood how much the adaptive immune response is involved, but that's a question that we are interested in. I think most of what people see in humans who have PAM is the preponderance of a very strong innate immune response. So the innate immune response, it senses that there is a pathogen and there's damage going on, maybe because the pathogen is causing damage, and it responds. It comes in, like a lot of cells come in looking for what's causing this trouble, and that's usually it's happening within the central nervous system in the brain of infected individuals, and that is going to we can talk about it later maybe, but it could be a good thing in some ways, but it probably plays a contributing role in ultimately the fatality of this of individuals, because your CNS, the central nervous system, is enclosed inside your skull, and you have there's only so much pressure that can build up inside of there, and if you have a really strong immune response, it creates a ton of pressure, and that by itself is pathological. So,
Sure.
people have asked me, is it the immune response is really bad, or is it the amoeba? And I think both of those things are bad in this context. It's just that they're kind of not misbalanced eventually, and that is hard to overcome.
So when we're talking about the immune response, would that be inflammation?
Yes, that's inflammation. So you have a lot of inflammation. When I say like you have a lot of immune cells that come into the tissues that causes inflammation, you get a variety of inflammatory pathways. But in general, yeah, you can call it inflammation.
Sure.
There might have been another part to your question. Well,
Oh no, you answered it all. Let me move on to another thing that I'm not sure if there's an answer to. Do we know if antibodies are produced when you are infected? And if they are they protective? Do we know anything of that?
Yeah, so with individuals who have PAM, well, very few of them survive. So that's very difficult to assess whether you made antibodies. And oftentimes people aren't checking, like clinicians and things like that. That's not their first priority to check whether someone's made an immune response, like a specific immune response to amoeba. In some cases, the time turnaround is actually kind of fast. So you're really not able to make a good antibody response fast enough before people actually succumb.
Right.
There have been people who have survived, and I think there is evidence from the CDC at some point had looked for antibody responses in maybe a surviving individual or even maybe a patient who eventually succumbed. And they were able to detect antibodies. And that's not surprising. The immune system is very good at making antibodies to things, and it can make antibodies to almost anything. So there's no doubt that your immune system could make antibodies against Naegleria. It's just whether it would have enough time to do it in a way that's protective.
Right.
That's, I think, hard to say. But that's not to say that individuals who are not symptomatic, like you and me, who may have been exposed to Naegleria, could have antibodies against Naegleria that do something that prevent us from getting infected. That's possible. So I think it's still a sort of an outstanding question whether antibodies could be useful against this pathogen. It just doesn't seem like in most cases these individuals, they're not able to make an immune response that has antibodies in it that's going to help them in some way. It's just when you progress to PAM, it's beyond the help of antibody.
Right. Do you know, or do you believe that there's asymptomatic infections of this parasite?
Yeah. So it's kind of a semantic question, because infection, what does it mean, right? Does it mean that if I have Naegleria in my body, am I infected? If you mean do people get PAM, or do they get amoeba across their upper airway and then not die? That I have no idea. I think that seems unlikely, but it's possible.
Yeah.
I think it's very likely that people are infected, through their cuts in their skin, or they swallow it or something, like they get it inside of them and enough to generate an immune response that generates antibody. And there's a lot of data actually that suggests that a lot of people have antibodies against Naegleria fowleri, which means that they had exposure to it. It either means, there's some cases, I guess, you could be exposed to dead amoeba, but in general, it probably means they got it in them. and it was alive briefly before it was killed, and then they were able to make an immune response to what was left of it, and now they have antibodies against that. But yeah, so asymptomatic, I mean, I think in general people think, is it possible to get amoeba up your nose and then not die? And I don't know the answer to that question, really. I mean, I would guess it's possible because most people don't die, and there's a lot of water going up people's noses. But what the factors are that regulate who gets really sick and who doesn't get sick at all, I don't know. I mean, it's also possible people get, sick a little bit and they don't realize they were so close to being really, really sick. Those are just hard things to know. I mean, people get headaches, right, after they go swimming, but you don't know what caused that headache. Could be anything.
Yeah, it's incredible how many unknowns there are to this parasite.
I'm sorry, I'll just interrupt you, but sure. I will say that in mice, we work with mice as a model organism, and in that case, their anatomy is a little bit different in their nose, but we don't really see asymptomatic infection. They either get infected or they don't. They're either going to get really, really sick or they're not going to get infected at all.
Now, you published a short clip of Naegleria in the brain, and I want to go ahead and play that and see if you could go ahead and try to narrate the 20-30 seconds worth and give the audience a picture.
Sure.
Okay.
Okay. I can preface this maybe a little bit by saying that, we use intravital imaging techniques that maybe you can play it twice, but I'll just describe what I'm talking about. So these are images basically that we take inside of a mouse that's infected with Naegleria fowleri. And the mouse is alive, it's sedated, obviously. And we're imaging through the skull, basically into the brain beneath the skull. And in this movie, which hopefully you can play again, we'll see that there are a lot of immune cells. Immune cells here are these green things or cyan colored things in the other movie. And they come into the brain like crazy. This is like the sort of description of what the response to amoeba infection is that you get a lot of immune infiltration. A lot of cells come in, this inflammation. And what you see is that there's a lot of cells that are very active, kind of roaming around. This is obviously sped up a little bit, but they're moving around like crazy because they would not normally be in the brain at all, but they're moving around like crazy. And in this video, you see this like little circle that comes up. You can see like a shadow right here where that is the amoeba, we believe, at the front end of this huge horde, a swarm of cells that are coming. You see another little circle at the bottom. These are all immune cells that are detecting the amoeba or maybe the damage that the amoeba causes, and they're chasing after it. And here you see it again at this little circle. At the front edge of this big surge of green at the front edge here is the amoeba. You don't really see it as well here, but you can see by the behavior of the immune cells that they're chasing something. They actually turn that corner and they keep chasing after it. And all these other clumps that you see in those images, those are areas where the immune cells have actually managed to encircle and capture an amoeba, and they just hold on to it there and kind of kill it within that big clump. So that's what these guys are trying to do, these immune cells. Like these huge masses that are following the amoeba, these are many cells, like hundreds of cells chasing after that amoeba. And they're trying to get we're not sure what they're doing exactly, but they're trying to either get out in front of it or get around it and circle it. Like here you see these big clusters at the bottom right and other areas with big clusters of cells. Those are all around one amoeba, and they've basically killed the amoeba inside of there.
Yeah, that's really something.
So this is a very dynamic process because these cells are these immune cells are designed to go to the site of damage or infection. And in this case, the damage is moving, right? Because the amoeba is moving around. So we're hypothesized it's possible that they're actually chasing the damage that they cause. But in the end, not always, but they will find those. They're capable of finding those amoebas and actually stopping them.
Yeah, well, thank you for that narration. I really appreciate it. It's quite a fascinating video. Well, there's so many unanswered questions, and I really want to learn more about the research that's being done at the Moseman lab. Dr. Moseman, what research projects are you and your team working on, and what questions are you hoping to find answers to?
Yeah, so regarding Naegleria, at least, We know that the immune response fails to get rid of amoebas in most individuals. So we discussed a little bit about asymptomatic, the possibility of asymptomatic infection, but and at least in people who are symptomatic, like PAM patients, the immune response is not able to clear the pathogen. And we have, again, we do all of our work in animals, and in mice, our studies, we can pretty clearly show that the immune response does do something. It's not like it doesn't do anything to stop the amoeba. It definitely slows down the process. So what we want to know is, what are the mechanisms that these innate cells, the ones we saw crawling around in there, what are they actually using to find the amoebas as they move around? And then what do they use to capture, sort of like encircle them? And then what do they use to actually kill them? Because they can kill them. They can lower the amount of amoeba that's there. And we really want to understand what those are, because we think that some of these the more inborn mechanisms, is not innate ones. Those are the really critical ones for the immune response to use to slow this and maybe prevent the pathogen from really getting established in the central nervous system. And we think there's a lot, we think we can understand a lot about what could be failing in certain individuals and maybe leverage that clinically to make better therapeutic strategies, let's say. And then you've already alluded to it, but antibodies, we don't really think that people who get infected are probably making antibodies that are protective, but that's not to say that you couldn't use antibodies to be protective. And one of the things that we're looking at is whether, if we use, specific antibodies that we've made in the lab that are extremely specific to Naegleria, if we can use those to target Naegleria inside, animals, or maybe even eventually in humans, can we direct the immune response to those amoebas in a way that allows for a more complete capture and clearance of amoebas, like accelerate and enhance the response that you're getting from the innate immune response, immune compartment, because those two go hand in hand. And we think that there's a lot of potential there, just because the antibodies are very specific. I don't think like a widespread vaccination campaign would be very good for a number of reasons, but the number of patients who are diagnosed with this is pretty low, and therapeutic options are bad and really don't lead to survival very often. So we don't have a lot to lose. And so we think that there are opportunities here to try to, take some different approaches, maybe using antibodies in a way that could help slow down things and allow the body to catch up in a way, even just anything to slow this process down and increase the chances of survival. And then I guess one other thing we're kind of interested in is, we also kind of alluded to this throughout this conversation, but what is like the normal sort of population level human response to Naegleria fowleri? So I can't imagine that I haven't been exposed. I mean, I spent a lot of time swimming in warm lakes as a young person, maybe not so much now, especially now that I work on Naegleria fowleri.
Exactly.
But I mean, I still go into fresh water, and I have to think that there is a lot of exposure that goes on and people just don't know about it. And are people being infected symptomatically just by happenstance, or is there something that's different amongst some people's immune response? It could be also a mixture of those two, but we are really interested in trying to look at the prevalence of antibodies in the human population, and not just antibodies, because I think one thing that people often forget about is, if you want an antibody to do something for you, it needs to be able to reach its target in a situation where it can be helpful. So a lot of antibodies get made to like internal things, so like inside the guts of the amoeba, let's say. But that's not something that antibodies would normally be able to see in an intact pathogen. I guess the analogy I was thinking of is like an egg. Like if you made like egg has an egg shell, right? You want to be able, like the amoeba is an egg, you want to be able to target the shell, because if it's alive, that's the part that you could get to. The inside part, it doesn't matter. Like you can make as many antibodies to the inside as you want, but it can't help you fight that pathogen because it can't access that part. It can only interact with the outside part. So that's one of the things that's important, is having antibodies to the outside part of the amoeba. Otherwise, those antibodies are useless. They're not going to help you at all in pathogenicity. So you know, if we look through human populations, maybe get some idea of some people make better antibody responses to the outside versus the inside of the amoeba, and that maybe gives them an advantage to, you know, prevent infection in the first place. But those are things that, you know, haven't really been done very thoroughly. There have been certainly studies of like amoeba reactive antibody in people's serum, and there's lots of it. It's just, what are those things reacting to, and could they be helpful? And it's not hard to do. We just got to be able to get patient cohorts and get people, or, you know, normal human controls, just control humans just to see what they look like, and screen those things. Those are things we're pretty excited about. We have sort of limited bandwidth on all we can do at the same time, but those are things we think are pretty tangible and could be useful, at least answering questions. Some of them clinically relevant, others just more like curiosities, like why some people get infected and others don't.
You have any estimated dates of publication for anything? No.
I wish, man. I wish.
Yeah.
No, we don't.
Yeah.
It's impossible to predict that kind of stuff. Also.
Yeah, I'm just interested. Sorry.
Yeah, no, that's fine.
Yeah. Okay. Well, let me go ahead and just close out. See if you had any final thoughts about the parasite itself, the immune response, the research, anything we didn't cover, what we should have covered.
Naegleria is obviously a pretty devastating pathogen when it becomes a pathogen. But I think what's interesting about Naegleria is it's not really what you would call an obligate parasite. It doesn't seem to want to be inside people. It just is this weird confluence of getting into the right spot and then being able to take advantage of some lack of control in people and in most animals. Honestly, lots of animals can get Naegleria fowleri infection through their noses, and that's so my lab is also interested in just immune responses at this sort of olfactory smelling surface. And that's what's interesting to us about Naegleria when we first got into, is that it's really only a pathogen when it can access this one surface. You know, if you inject it into your arm, it doesn't do anything. Your body just kills it. But there's something about this surface mixed with the pathogen that we think is kind of useful in shedding light about how immunity works really at these olfactory surfaces in general. And we think that it's a nasty bug, but it also is kind of useful in like showing exactly the vulnerabilities that these surfaces have. But yeah, our research is, we're doing a lot of different things about Naegleria and other things about these olfactory surfaces to trying to understand how immunity and barrier function works up in the upper airway, especially this olfactory surface.
Good stuff. I really appreciate you coming on today. Very interesting topic. And I want to thank you, Dr. Ashley Moseman, for sharing your time and your expertise. I really appreciate it, sir.
You got it. Thanks a lot, Robert.